Good afternoon. Welcome back to Virology. Today we start the second half of this course where we're going to discuss how viruses cause disease and how we can intervene. And today, some fundamentals about how infections, viral infections, take place. Now, so far in this course, we have talked about everything we've talked about pretty much has been done in cells and culture. Here, this culture dish full of cells, we've learned about entry and transcription and DNA replication and RNA synthesis and so forth, all from cells and culture. But of course, infection of cells and culture contributes zero to the maintenance of virus in nature, right? Because it's in a laboratory. What we're going to talk about for the rest of the course is infection of hosts, where not only must the virus infect the host and make more viruses, but it has to get out and find another host. So you have to get in and out, and if you don't do that, you're finished as a virus. And most likely over time, many viruses have come and gone because they can't find a host. It doesn't help to go in a host and stay with that host until the host dies. You don't perpetuate. Remember, we live and prosper in a cloud of viruses, and in fact, most encounters with viruses have no consequence. We say that many infections are either inapparent or asymptomatic. All right, so those are interchangeable terms. And let's define what is asymptomatic. What does it mean? A symptom is what only you can feel. So, a stomach ache or a headache or fever, I guess that could be measured. Anything that only you can feel. That is a symptom. That's the definition of a symptom. So an asymptomatic infection is one where you feel nothing. Doesn't mean things aren't happening, but you don't feel it. And the danger is you're walking around society shedding viruses and infecting other people. So symptoms apparent only to the patient. Then we have signs of infection. A sign is something that could be observed by others. So the rash of measles is very obvious. It's a sign of infection. Fever, you could measure. You could measure viral genomes in blood by PCR. So all of these things that you can measure are signs of infection. And even though you may feel asymptomatic, if a virus is in you reproducing, you certainly have signs, but often the signs are not obvious to you. All right, very important distinction, symptoms and signs. Now, here's an example of asymptomatic infection. West Nile virus was absent from the Western Hemisphere until 1999. It was found elsewhere. It arrived here in New York City in the summer of '99. It arrived in Queens and began to infect people. It's a mosquito-borne flavivirus, and the mosquitoes that transmit it are here in the US. And in 4 years, it spread across the US. So here are maps of the incidence. So 99, New York State, and then slowly expanding through the years until it was across the country by 2008. Now it's throughout the Western Hemisphere. Now, by October of 2004, there were about 1 million people infected in the US. And when you see a number like that, that can only be derived from either a diagnostic test, which we don't do on a large scale for most viruses, or serology. You take blood from people and you look for antibodies against the virus, and you say, aha, you've been infected with West Nile virus. And then you ask them, have you ever felt any of the symptoms of West Nile infection. You don't say signs because they don't measure signs, they feel symptoms. And 20% of the people said, yeah, I had febrile illness back then, which was probably West Nile. But 80% of people felt nothing. So they were asymptomatically infected. And about 1% of the people, the virus gets into the central nervous system and causes severe neurological disease. There's a good reason not to get West Nile infection. Of course, now you are young and you will not be predisposed to CNS infection. But as you age, the incidence goes up. And we'll take a look at that later in another lecture. By the way, we haven't talked about this yet, but I'm going to throw out this tidbit. Every West Nile infection of humans is a zoonotic infection. That means the virus goes from animals to people. Every West Nile infection, a mosquito has bitten a bird or a rodent, picked up the virus, and gives it to humans. No, there are no cases where a mosquito bites a human and then transfers the virus to someone else. The viremia, the virus in the blood isn't high enough. But I digress. We'll get back to that later. So obviously, 80% of people were asymptomatic. They had no obvious disease. This makes it hard to stop an epidemic, right? SARS-CoV-2, that was the big problem with SARS-CoV-2. Many, many people were asymptomatically infected. And there was also another issue as well, which we'll talk about today. It was impossible to stop the spread of infection. Fundamental virology. It's been here for years. It's nothing new with SARS-CoV-2. It rhymes. How about that? All right, another concept you need to know, because you're going to know it, and most people in the world are not. Most people get this wrong. What's the incubation period of a viral infection? It's the period before symptoms occur. It's the period before you feel stuff, right? Obviously, the virus has already gotten in you. The genomes are replicating. The host is responding, immune responses, as we'll see in a couple of lectures. So these are signs. Signs are present, but no symptoms. That's the incubation period. And you can see there are viruses multiplying there. You get infected viruses. So these are signs. You can't feel them, but you could measure them. And then, boom, one day you start coughing and sneezing and sore throat. And that's the end of the incubation period. You now have disease. And I cannot tell you how often this happens on subways and trains. People just sit there coughing away with no concern about anyone else. No masks. I don't understand what they're thinking. Try to be sensitive that if you're coughing, you're going to spread viruses to other people, and maybe they don't want it. You know, there are a lot of things people don't want from you, right? They don't want diseases from you, and this is one of them. So be careful, think about it. Okay, the other point that's very important is whether the virus is transmitted during the incubation period. Because if you're walking around with no symptoms and you're shedding virus, boy, you're a big risk for infecting other people, right? If someone's coughing away, I know to get away from them or put on a mask. But if there's not doing anything, like some of you could be shedding virus. You could be asymptomatic. We wouldn't know. And it depends on the virus, all right? And we're going to get back to this later. So these incubation periods vary for different viruses. They range from short, so influenza virus, a day or two, rhinoviruses. Ebola virus can be 2 to 21 days, and all the way up to 50 to 100 days, 50 to 150 days, which is the incubation period for warts. You know, the warts that you get on your feet mostly, but sometimes on your hands, those are caused by viruses. They're called papillomas, and they're caused by viruses. Hundreds of different kinds of wart viruses. There are sexually transmitted wart viruses that cause cancers. They can cause anogenital warts, but they can also cause cancers. Those would be the human papilloma viruses, and we're going to talk about those later. These are the viruses that RFK Jr. is trying to minimize. Okay, if you get vaccinated against HPV, it will prevent you from getting cancer. About 14,000 deaths a year in the US pre-vaccine. Yet he says, we don't need them. Can you imagine how this drives me crazy, having done this for 44 years and learned the facts, and to have a guy just walk in and lie and people believe him? When you finish this course, you're not going to believe a word he says. Maybe you don't already. Anyway, incubation period, short, mean typically the virus is causing disease at the site of entry in the host. Long, the virus has to go somewhere. SARS-CoV-2 didn't make this table, 5 to 7 days. And there's also another term called the prodrome, and that's the period of symptoms before those that are characteristic of the disease. So say, for example, measles virus, the rash would be characteristic of the disease. So anything before that would be the prodrome. It's just a word you might encounter. I'm not going to use it anymore, most likely. There are a bunch of terms that you encounter in when you talk about viruses causing disease. And, onset of COVID, nobody knew how to define these or understand them. So I started including them in this course. And the ones I'll tell you about: morbidity, mortality, incidence, fatality. So here we have pictures of people. Each icon is 10,000 people. The red or the orange are PCR positive, so they're infected. And then the red are people with signs and symptoms. They're also PCR positive. And so the incidence, as you can see, is the number of people infected just divided by the number in the population at a particular time. The morbidity rate is the number of people ill over the number in the population. The mortality, number of deaths over number in the population. Very straightforward calculations. The case fatality rate is the number of deaths over the number of confirmed infected. All right, so here we have a mortality. Here's the mortality rate of 5%, and a CFR 10,000 over 50,000 would be 25%. And finally, the infection fatality rate is the number of deaths over the number of actual infections. Now, the problem is you never know the number of infections during an outbreak. You can't test everyone, right? So you're always making extrapolations. And the infection fatality rate is an attempt to use statistical procedures to actually gauge the number of infected people based on PCR positivity or some other assay that you're using. Case fatality rate was widely misused during COVID pandemic, all right? Let me show you why you cannot say much from the case fatality rate. So here's the first 44,000 cases in China, reported in China's CDC Weekly. And total cases, 44,000, deaths 1,000, so that's a case fatality rate of 2.3%. So here we have a defined population where we know who was infected and we know who died, right? But look at, if you stratify it by years, young, hardly any deaths, right? And then as you age, you get more and more deaths until over 80 years of age, 15% case fatality rate. Amazing, right? Age influences it. So does sex. Men are twice as likely to die of COVID than women. Where you are makes a difference. Here's a graph of different places in China at different times during the outbreak. And so Wuhan, of course, where the infection started, in January, 20% case fatality rate. Then with time, it drops. In China overall, similar trend. But if you look to Hubei outside Wuhan, and in China outside Hubei, very little case fatality. Why is that? Well, Wuhan was overwhelmed in the beginning. The hospitals couldn't take care of people. So if you can't bring someone to a hospital, they're going to die. The CFR is going to go up. That has nothing to do with the virus. It has to do with hospitals. CFR is always iffy in my view. And outside of Wuhan, plenty of hospitals, not that many infections early on anyway, so you had a lower fatality rate. Now, I'm telling you this because the press will say, oh, a new variant with the CFR that's higher. It means nothing. It means nothing unless you know what exact population is it, right? Are they old? Are they young? Are they immune? After a while, you have to look at immunity, yeah.
How does comorbidities get calculated into this?
That's what age does. You're likely to have more comorbidities. So aging, one of the major ones is making antibodies to interferons. We'll talk about that later. And having a weak immune response, which happens as you get older. But asthma, cancers, diabetes, all these other high blood pressure, they all make the CFR higher. So it makes zero sense. Look, 2.3% overall, but if you're over 80, it's 15%. So it really matters. But still, the press doesn't get it, I think. Many people just use this CFR. If you want to use something, use the infection fatality rate. It's a little better estimate of the overall fatality rate. But it still has to be stratified by group. So our first question, which of the following parameters is not influenced by human interventions? Mortality rate, case fatality ratio, infection fatality rate, incidence, or incubation period. All right, looks like we've stopped. If we do, 100%. Lecture 12, right? It's your first 100%. This is a 100% lecture. Some of these are very straightforward, but also make sense. The incubation period, it's the virus properties cause the incubation period, but all these other things, the mortality rates, incidence, as I've been going on for a few minutes, they can be influenced by human interventions. They're not a property of the virus only. Omicron is mild. Really, in who is it mild? In young people who are already immune? Of course it's going to be mild in them. Any coronavirus would be mild in them. It's not mild in old people. Since Omicron, we still have about 1,000 deaths per week in the US. Is that mild? It's pathogenesis. So for the rest of the lecture, let's talk about some principles about how viruses cause disease. Pathogenesis is the production. Well, pathogenesis is the process of producing a disease. We have viral pathogenesis, we have bacterial pathogenesis, all kinds. And we have viral pathogenesis, which we talk about in this course. There are two components to this. You have to remember this. There's what the virus does to you. It gets in and it kills cells and so forth. So if you kill brain cells, you're going to have brain issues, right? But then there's also the host response. The virus gets in and you have immediately a host response that we're going to talk about later. And that can make a big difference. It can hurt you. In fact, many virus infections, all of the disease is caused by the immune response. We call that immunopathology. We're going to talk about that later. So two components, the virus and the host. In the last 20, 30 years, virologists have turned to studying viral pathogenesis. So all the stuff we talked about in the first half of the course, hardly anyone does that anymore. And most people focus on disease. How does the virus get in the host? What's the host response? Where does it replicate first? How does the virus spread? What organs and tissue are infected? How does the host respond? What kind of interferon antibodies, T cells, et cetera? Is it cleared? Or is a persistent infection established? All of us have herpes viruses in us that we acquired at a very young age. That's a persistent infection. Can't get rid of it. And finally, how does the virus transmit to another host? There are many ways. So we're going to cover these today. 3 requirements for a successful infection. You need enough virus. In most cases, 1 PFU is not enough. But for norovirus, 10 PFU is actually enough. We know that because we feed volunteers different amounts of noroviruses. And we can do that because they get better. They don't die. We make sure they're hydrated. There's down at Washington University Medical School, on Friday they recruit medical students. They feed them norovirus. They puke and vomit and diarrhea all weekend. And then Monday they're fine. They go back to class. And you can make a lot of observations from that, including if a vaccine works or not. Anyway, you need enough virus. The cells have to be accessible to virus. They have to be susceptible. and they have to be permissive. Those terms still apply here, susceptibility and permissivity. And local antiviral defense is either absent, there's some places where there's no defense because it would hurt the tissue, or it's overcome. The virus replicates so fast, it overcomes the local defenses. So you have an older person whose interferon response is bad, the virus will overcome it. Whereas younger people tend to have better interferon responses, but not everyone. Everyone is different. Each one of you is genetically different. So you're going to respond slightly differently to any infection. All right, let's talk about getting in. The body has only a few places where viruses get in. It's pretty well protected, and they are shown here. There's the skin, there's the mucosal tracts, the respiratory, alimentary, your genital tract, the conjunctiva of the eye. Not much else. So let's cover these in some detail. The skin, the largest organ in your body, the skin, right, by weight. It has an outer dead layer. So the outer layer of your skin is called the epidermis. The outer layer is dead, and it flakes off periodically. And the dust you see floating in rooms is your skin in part. When you see the sunlight flowing through a window and it's speckly, it's your skin, okay? Sorry, it's the truth. That's a great defense against infection. Viruses cannot reproduce in dead cells, right? So viruses fall on your skin. They're not going to reproduce unless you have cuts, which we all do, or if a dog bites you, or if a mosquito bites you. If viruses can get below the dead layers, then they can reproduce in the living cells below. And there are blood vessels and cells, lymphatic vessels, all kinds of cells down there. And if the viruses get down there, they could get into the blood vessels and spread. We'll take a look at that. But mosquitoes are a unique vector. So the way mosquitoes transmit viruses is they take a blood meal, and only the females take blood meals because they need blood to make eggs. The men eat sugar water. They eat sap from trees. So they don't transmit infection. But the females just find a host to get blood, and if that host happens to be infected, they're going to pick up virus along with the blood. And what the mosquito does is amazing. You should go look at this video I made. It lands on you, and then it puts its proboscis through the epidermis into the underlying tissue. It looks for a blood vessel to stick its proboscis in. It probes and it probes until it gets in one, and then suddenly the whole network of blood vessels collapse as it pulls out blood. And of course, this probing, if the mosquito has viruses in it, the probing is releasing viruses from the previous host that the mosquito had a meal with. And along with probing, the mosquito is depositing anticoagulants, so you don't clot up and it can take more blood, and also painkillers, so you don't feel it and slap it away. So it's going to be sitting there, maybe on your back, unbeknownst to you, until it can get blood. So you would need at least 2 blood meals. Mosquitoes don't live longer than that. They don't usually take more than 2 blood meals. But if you can get 2, the first blood meal, you pick up virus. The second, you deliver it. That's how viruses are transmitted by mosquitoes. But there's, of course, also dog bites, and needle sticks and scratches can all deposit virus through the skin. All right, so that's the skin. Pretty straightforward. Mucosal surfaces, we have lots of these in our mouth and nose and the conjunctiva. Respiratory, alimentary, urogenital tract, they are lined with living cells. They have to take things in and give things off to the environment. So actually, they're all exposed to the environment. So basically, even though there are valves, these are considered external, and you have air going through them, and of course, whatever you eat as well. And so these are ripe for infection. They're kind of a vulnerability, because if you had dead cells lining this mucosa, you wouldn't get infected very easily, but then you wouldn't live long because you couldn't take in nutrients and get rid of things, right? So the upper tract, the respiratory tract looks like this. The whole tract, there's an upper and a lower tract, and they're joined, of course, by the trachea. And they're lined with epithelial cells of different sorts. Some of them make mucus, like the goblet cell. So there's a layer of mucus over these cells, and they extend throughout the upper tract, and all the way down into the lower tract. Now, the upper tract is kind of a catch-all, right? Viruses come in to the upper tract and it gets trapped. And they may replicate up there, but if your immune system takes care of things, they stay there. Otherwise, they can spread to the lower tract and cause serious problems. So here are some of the viruses that will infect these two sites. And on the right, the clinical manifestation. You know, this is all depending on what location the inflammation is happening. Rhinitis, pharyngitis, laryngitis, all the way down to bronchopneumonia. When particles go all the way down into the lung, it's not easy to do that. Viruses initiate in the upper tract. You breathe in and you pull viruses in, they're replicating in your palate and all that mucosa, turbinates and so forth. And if the infection gets out of control, then the virus can start going down into the lung, and that's serious. Now, the tract is lined with ciliated epithelial cells and mucus, and so these are always, as you move down into the tract, these are always beating upwards to get rid of particles that are trapped in the mucus. So to a certain extent, the lower tract can clear things that come in. But as you get older, this cilial activity gets worse and worse. It doesn't work so well. So you have more and more viruses going down there. Also, as you get older, you tend to aspirate more. You tend to swallow, you inhale saliva, right? Which doesn't make any sense to you now, but I guarantee you it's a thing. Every now and then you'll inhale a little saliva, it goes down into your lungs. And if you've got an infection, you're putting viruses down there. I heard a ENT guy at Penn talk all about this, that's a big deal, this aspiration. And so most infections are localized to the upper tract, but once you get down into the lower tract, then you have serious issues, because then if virus is multiplying, and more importantly, the immune response is kicked in, and you have inflammation and you can't breathe. You have an interference with taking up oxygen, and that's when they put you in the hospital, in the ICU, and intubate you, and that's a big problem, because intubation is really bad. Okay, that's the respiratory tract. And we have the alimentary tract, of course, beginning with your mouth and going all the way through to the anus, and that's lined with mucosal cells as well. So here is the small intestine as an example. Here's a cross section. We have the lumen of the gut here, and then we have the epithelial cell sheet. And these cells are all cells with villi, as you can see here. And then we have underlying cells as well, and eventually muscle and connective tissue and so forth. So these are polarized epithelial cells. They have an apical and a basolateral domain, and they're in contact with the outside world, right? You eat things and drink things, and they pass through here, and a lot of viruses can go through and be taken in. And there's a huge immune system in the gut that monitors foreign antigens, because probably gets more in that sense than the respiratory tract. You know, the respiratory tract samples about 6 liters of air a minute. That's a lot. 6 liters of air, just think. And so you can bring in a lot of particles that way, but the gut is even more, I think. Anyway, here's an example of the immune system. This M cell is actually part of the gut immune system. There's a lot of lymphocytes associated with it. Macrophages can get really close to it, and they can sample the antigens that are in the gut to make sure that they're not something that's foreign. And many viruses can pass through M cells. Here's an EM of some viruses binding to M cells and passing right through. So viruses can infect these cells. And an important point is that at the bottom of the cells is what's called a basement membrane. So it lines the bottom of these enterocytes. So that would be here in this picture. So there's a basement membrane, which is a permeability barrier. It prevents things from just diffusing through. As you can see here, immune cells can get through this. And when they do, they break it, and that's when viruses can get through from the apical side. So when you get infected, you draw immune cells to the basement membrane, and they break it, and viruses come through. Otherwise, they wouldn't be able to go through. We'll come back to that. I'll show you that in some more detail. So then we have the urogenital tract, also lined with epithelial cells, mucosal epithelial cells with mucus, and you can get infected there from sexual activity that causes abrasions. Some viruses will multiply locally, like human papillomaviruses, but you can acquire HIV and herpes simplex viruses this way, and they will spread to other sites, as we'll talk about later. The eye is another portal of entry. We have many complicated cell layers here, but the outer layer is composed of epithelial cells, and in particular, conjunctival epithelial cells line the inner eyelid. And there's a thin layer of those on top, and then they go to the top eyelid as well. And all of these can be infected with viruses. You can see the viruses below that infect these different parts of the eye, and mostly on the front here, obviously. We'll talk about the vitreous cavity later on. This is a very interesting place as well. Now, if you ever have this situation where you have bleeding in your eye, that can be physical, like high blood pressure, but viruses can do that as well. They break blood vessels, and it looks very scary. All right, that's the eye. And of course, viruses can cross into the fetus from the maternal circulation. And there are two ways that this happens. There's transplacental, crossing the placenta, right? So here's a schematic of the maternal fetal interface. So you have fetal circulation abutted to maternal circulation. And here are our syncytiotrophoblasts, right? They're all one cell with many nuclei. That's made by a former fusion protein from a retrovirus that was stolen. We talked about that. And then there's perinatal, around birth infection. So it's different. So if there's a lot of blood during birth and the blood has virus in it, it will infect the newborn now. So these are called torch pathogens. Years ago, they were given this acronym, including toxoplasma, a parasite, rubella, cytomegalovirus, HIV viruses, and then they said other because they figured there would be more. And in fact, in 2015, Zika virus was identified as a virus that could go, from maternal circulation to fetal. It crosses this barrier because they have special properties, and now they infect the fetus and cause problems there. Next question is, the outer layer of which of the following is dead but can still serve as a portal of virus entry? Respiratory tract, alimentary tract, eye, skin, urogenital tract. All right, let's have a look here. Okay. Yep, 100% skin. Good. Skin is, outer layer is dead. None of these other things are dead, of course. Fortunately for you and us. All right, so we've gotten into the host. How does virus spread from the site of entry? So many viruses don't spread very far. They stay localized, and they can spread. So here's an epithelium with a mucus layer, and the viruses can infect these cells and then just spread locally in the epithelial cells. And other viruses can spread away from this. We call those disseminated. And if the virus infects many organs, we call it a systemic infection. In order to get away, both physical and immune barriers need to be breached. So that's that basement membrane that we talked about. That's a great barrier to virus spread. And when viruses infect cells, they cause inflammation. Inflammation is the migration of an immune response to the site of infection. We'll see how that happens later on. But if these cells are infected, for example, immune cells will be recruited to this area, and they will break down the basement membrane so that they can get in and sample what's going on there. And inadvertently, they let the viruses go through the basement membrane and spread systemically. Now, viral spread also requires another property, and that is apical release. So these epithelial cells all the mucosal epithelial cells that we've been talking about, they're polarized. They have a top and the bottom. That's not always the case for cells. And there's an apical and a basolateral domain, a top and the bottom. So in order to spread to other hosts, viruses have to be released apically, right? If they're not released apically, they're not going to find another host. So apical release facilitates dispersal to other hosts. And we see influenza viruses budding apically, measles viruses budding apically, right? They're spread by the respiratory secretions. But on the bottom, vesicular stomatitis virus is budding from the bottom of the cells, basolaterally. And that's a property of the virus. So I want to give you an example of a virus where this could be changed. So Sendai virus is a virus of mice, which we use to study because it's a convenient model. And the wild-type virus, when you inoculate it into the upper respiratory tract of the mouse, they get a localized infection and they're fine, they recover. You can make a 1 amino acid change. So that virus, that wild-type virus, buds apically from the respiratory tract cells. You can make a 1 amino acid change in the spike glycoprotein of Sendai virus. It will now bud both apically and basolaterally. And now when you infect mice with that virus, the virus spreads systemically because it gets out the bottom, past the basement membrane, and into the circulation. So 1 amino acid can change whether the virus goes to the top or the bottom. So to spread, and you not only need to get out of through the basement membrane, but you need to be released from the basolateral surface as well. Okay, so here is our epithelium that the viruses are. This could be respiratory or alimentary tract, doesn't really matter. Here's our basement membrane. So viruses are infecting the epithelium and they're causing inflammation, and the basement membrane is compromised and the viruses pass through the basement membrane. Now what they encounter are lymphatic capillaries. Remember, the lymph system is collecting fluids from your tissues and bringing them to lymph nodes so that they can be sampled by foreign antigens. And so there are lots of lymph capillaries and they're very permeable, so that they can pick up lots of material, and not just fluid, but antigens. So viruses then move into the lymph, they go to the lymph node. Some viruses reproduce in the cells that are in the lymph node. If not, you can pass through the lymph node and then you get into the blood. So you get into the blood circulation and you could go anywhere now. And that's how viruses cause systemic infections. There is a distinction here that many viruses could get through the basement membrane, but they're not infectious. They're not reproducing anywhere else. They're just viral particles, which you can detect in the blood, but they don't play any role in the pathogenesis of infection. Now, when you get virus in the blood, it's called viremia. And here's an experiment showing you what viremia is. We're looking at days after infection. We've inoculated mice in the tail vein with some virus, and we measure the virus titer here. So just after the inoculation, you can see there's a big jump in the amount of virus. That's the virus you've put in, right? The time point is taken immediately after you inject. So this is passive viremia. It does not represent any reproduction. That goes down quickly. The virus circulates in the blood. It reproduces in some tissues and goes back in the blood. So now you have a primary viremia, which further spreads virus to other sites. And then you get a secondary viremia, which is much bigger because it's now reproducing in different tissues. So viremia is a problem because it spreads infection to, or potentially spreads infection to other tissues. But it's not a feature of many virus infections. But it's more of a problem for the blood supply, or it's an equal problem for the blood supply, right? Our blood is checked because we don't know how to make blood synthetically yet. So we have to have donors, and we have to check the donors to make sure they don't have certain viruses that are pathogenic. And if the blood has those viruses, we throw it out or use it for research. So that's viremia. Now here's a virus infection of mice to kind of put all of this together for you. This is a natural virus of mice called Mousepox. It's related to smallpox virus, but it's not dangerous to humans, so we can study it in mice. So here's an experiment where on day 0, we inoculate virus into the foot pad. So here's the foot pad. And so this is how mice acquire the virus in the forest. They're walking around on the forest floor, and their foot pads are broken, right? It's a tough life out there for mice. They're broken, and viruses can get in. So other mice have deposited viruses in the leaves and everything, and they pick it up. So this is a kind of approximation of that we inject virus into the foot pad, okay? And that virus initially reproduces in the skin. We've injected it into the foot pad, so now it easily gets into the lymph system. It gets into the blood, makes a primary viremia. It seeds spleen and liver, reproduces there, makes a secondary viremia, and that brings the virus to the skin, and the mice get this severe rash, all right? So it's kind of it shows you how virus can spread from the skin to other organs. Now, at the time of this all happening, the foot pad is swelling because there's inflammation, immune responses there, and eventually the rash appears after the secondary viremia. Viruses also cause rashes in people, many different kinds of rashes, and they have different names for them depending on how they look: maculopapular, vesicular, etc. Here's measles, which is in the news. I believe that's this one down here, a maculopapular rash, and then Coxsackie virus, measles, parvo, rubella, VZV, Zika virus all cause different kinds of rashes. Perhaps here's chickenpox. I had this as a kid. It was miserable. And here's smallpox, which is really, really bad. Good thing these people, if they recovered, they would be completely disfigured from this rash, which covers all their body, not just their hands. It's a good thing it was eradicated. Next question. Which of the following assist in viral dissemination in the infected animal? Viremia, basolateral release from epithelial cells, movement through the lymphatic system, inflammation at the basement membrane, all of the above. There's no evolutionary reason for viruses to make you sick or kill you. All they need to do is reproduce and move on to another host, right? So disease is an accident, the way I view it. It's an accident of the virus killing some cells and also the immune response, right? So for example, poliovirus infects your intestines. 99% of the time, that's all you get. You don't get any symptoms. 1%, it gets into the CNS and it causes paralysis. So it's an accident. And the virus, once it's in the CNS, it's a dead end. It's not going anywhere. So that's how I view it. All the diseases are accidents. It's not that the virus, people say that viruses want to make you sick. This is just nonsense. I don't want to do anything, right? Yeah, but you can manipulate models to figure out how the disease is caused, and that's valid because maybe you can prevent it, right? Hey, what did we do? So the answer is all of the above. All of these things assist in viral dissemination. Viremia, basolateral release, right? Movement through the lymphatic system, inflammation to break the basement membrane. So we've talked about viruses spreading through the basement membrane, through the blood. Viruses can also spread in nerves. This is called neural spread. Nerves have very extensive transport system because, neurons can be huge. They can be meters in length. So here, for example, is the skin, which is obviously innervated with sensory nerves. And viruses can get in there and be transported by retrograde transport all the way up to the sensory neuron. And you can do the same with muscle. Viruses can enter nerve endings in muscle. These would be motor nerves, of course. And the viruses can go up all the way to the motor neuron and destroy those as well. And this happens with a number of different viruses. So this is neural spread within nerves. And it's done by motors. here's a neuron shown here, a cell body, and the termini where synapses occur, right? And within this long axon is microtubule structures that have motor proteins on them. Kinesin and dynein, they go in different directions, and often the virus particles will be moving either away from the cell body or towards it for infection. People have used this to map neural circuits because viruses can move along nerves. So they take a virus, label it with GFP, green fluorescent protein, inject it at one place very specifically to one neuron, and then over time they can trace the GFP and establish all the synaptic connections that are occurring. And now this is used extensively in neuroscience research to target specific neurons, to knock them out or knock genes out and so forth. So viruses play a huge role in neuroscience research now. If you just read any contemporary paper, amazing what they do with viruses. Now, infections of the CNS have some specific terminology. Yes?
If your neurons get infected, is your immune system, like, I know they're like microglia and other stuff, but is the immune system more hesitant to like attack it because you don't get new ones?
Yeah, so neurons don't have MHC, so they're invisible to T cells. And it's for that reason. But there's still immune responses that happen in the brain. There are microglia, and then some cells come in from the periphery as well. Yeah, and it causes damage, for sure. I mean, it is well isolated, but it's not perfect. So a neurotropic virus simply means it can infect neural cells, no implication about how the virus gets there. A neuroinvasive virus can enter the CNS after you put it peripherally, like in the leg, it can enter the CNS. That's neuroinvasive. And neurovirulent means it causes disease of the CNS. So 3 examples: herpes simplex virus, low neuroinvasiveness. So if you have a herpes simplex infection, it's very unlikely it's going to get into your brain. But sometimes it does. And if it does, it's very neurovirulent and can cause encephalitis, herpes encephalitis. About 1 in 250,000 cases of herpes in the US. Mumps has high neuroinvasiveness. So back before we used to vaccinate against mumps virus, which caused the rash disease in children, very much like measles, it entered most kids' brains. There was evidence that in most kids the virus got in the brain, highly neuroinvasive, but it didn't cause any disease. So low neurovirulence, high neuroinvasive. And then rabies is the worst. Highly neuroinvasive. If you get bitten by a rabid animal, the virus is going to get into your CNS. And once it's there, you're dead unless you're vaccinated. Almost 100% fatality with rabies virus. High neurovirulence, high neuroinvasiveness. That's the virus I'm the most worried about, because there's a lot of rabies left in the world, not much here in the US, but in India, there are 70,000 cases a year of dogs biting people and getting rabies. Lots of rabies. All right, so we're in nerves, we're in blood. How does the virus get into tissues? Tissue invasion. There are 3 different ways, as shown here. So in some tissues, and they're listed here, the blood is really tightly isolated from the rest of the tissue. So this would be like the CNS. And this is a capillary, right? So there's an endothelial cell forming the capillary wall. And there's no junctions in the capillary. And then there's a basement membrane here. So really hard for viruses to get across. It's not impossible. They can do it, but they're impeded. This is why it's not easy for viruses to get into the CNS. Then the next set are like intestines, kidney, and so forth. You have a basement membrane, but then the endothelial cells have pores. So here we have 2 endothelial cells making up the capillary, and they have pores, so viruses can get through it. If they can cause inflammation, they'll get through the basement membrane as well. And then finally, on the left is the liver and some other tissues as well, where there's not even a capillary, it's just a couple of endothelial cells lining what we call a sinusoid. and the blood just flows through. So your liver, this is good for your liver because the liver is filtering out things from the blood. So blood has easy access to the hepatocytes. But for a virus, everything you put in the blood goes to the liver first. And so viruses can easily get through those gaps and infect the hepatocytes. That's why we have so many viruses that infect the liver. But that's the structure of the liver to allow it to function. Now, but viruses do cross these tough situations, right? So here are some summaries of how that happens. This is a capillary with a basement membrane around it. And the capillary, you can see here, so lymphocytes and monocytes can break the basement membrane. So if they have viruses in them, which happens, they will take the viruses out of the capillary into the surrounding tissue, right? Some viruses reproduce in the endothelial cells and cause inflammation and breakdown of the basement membrane and get through that way. And other viruses are brought through by what's called transcytosis. So they're brought up by endocytosis into the endothelial cell and then deposited on the other side. They're not infecting these cells, but this is a way to get across. Now, the CNS is very well protected, as I've told you, but viruses can get through in various places. Most of the capillaries, in addition to having these tight junctions in the endothelial cells and a basement membrane, which is colored differently here, but it's there. They also have astrocytes, like making an armor around the blood vessels, right? To completely try and keep viruses out, but it doesn't always succeed. Many viruses can get through this. And here on the left, there's certain ways that viruses do get into the CNS. So they can get in from nerves. Remember, don't forget that. They can be brought in from various nerves, even the spinal cord. In the brain parenchyma itself, the main tissue area of the brain, viruses can get out of capillaries, as I told you, by causing inflammation and so forth, as I summarized on this slide. And then the meninges has blood vessels in it as well, and viruses can get out of them and travel from the meninges into the brain parenchyma. The CSF is made in a part of the brain here, which has very loosely, the choroid plexus, there it is. I was looking for the name of it. So the choroid plexus makes CSF, cerebrospinal fluid. And the blood vessels there are pretty permeable compared to the rest of the brain, so viruses can get out. They can go into CSF and spread elsewhere or get into the brain tissue as well. So this is how viruses get into the brain. All right, so there's our infection. We've gotten to different places. What happens in each of these places we'll talk about later, talk about some specific diseases that happen to illustrate it. But now we have to transmit to another host. We have to have, in order for a virus to propagate, it needs to spread from one host to another and do what we call is maintaining the chain of infection. Because if it doesn't, it stops, and that's the end of it. And transmission is influenced both by the virus and the host. So there's a parameter called R0 that tries to encapsulate the host properties and the virus properties together. So there are two general patterns of transmission. And it's not just humans, but everything else on the planet. We can go animal to animal. So here we have human to human, right? This is all our respiratory infections, our noroviruses, HIV, et cetera. It goes from person to person, nothing intervening. And then we have vector transmission on the right, from animal. A vector brings virus from animal to animal. We talked briefly about this before for West Nile virus. But here's an example of a virus that's spread among rodents by ticks. So the tick bites a rodent, takes a blood meal, takes virus in with it, and then eventually it will bite another rodent and give that rodent the virus, and this occurs in a cycle. And this is happening out in nature all the time. We don't study it very much. But happening a lot, and you go in the woods and you get a tick on you, it can have some pretty nasty viruses, and we're going to talk about those that will infect you and make you sick. So, and that doesn't have to be a tick. It could be a mosquito or any other kinds of vectors. With transmission, we also have terms that you should at least be familiar with. So, horizontal transmission. Transmission between members of the same species. So here we have some animals, sheep, I guess. And it is sheep, but these look like goats on the bottom. I don't know, maybe they're sheep too. They're non-human animals. And one sheep is infecting another, and that's horizontal transmission. If they were different species, if a sheep were infecting a cow, that would be a zoonotic infection. So zoo, the word for animal, that's from different species. So when we got SARS-CoV-2 from an animal, that was a zoonotic infection. Then it became a human infection because there were no more animals involved, right? West Nile is always a zoonotic infection. Every mosquito that bites you has picked the virus up from a rodent because it doesn't spread from person to person. So it's always zoonotic. HIV was originally a zoonotic infection from chimpanzees to humans. It became a human virus. Now it's no longer a zoonotic infection. Okay, vertical is mother and child. Here we go. Mother and child, vertical transmission. Iatrogenic. A healthcare worker gets you infected, something a healthcare worker does. And nosocomial is when you get infected in the hospital or a healthcare facility, which happens way more than you know. It's really a big problem. Healthcare-associated infections. And finally, germline transmission. Right? The agent is transmitted as part of the genome. Proviral DNA of retrovirus is integrated. It can be transmitted to offspring. Those are the transmission terms that I'd like you to know. Now, in many cases, virus shedding from the infected host is needed for transmission, right? In the form of respiratory droplets, skin lesions, blood. You can have a cut, and you're wrestling someone. Okay, that happens. They get your blood, or you have lesions on your skin. There's a herpes disease called herpes gladiatorum. Do you know what that means? Wrestler, because wrestlers get it. They have lesions and they wrestle and they give it to the next person. And urine, semen, feces, these all contain virus, so they can transmit infection. So respiratory droplets, influenza, SARS-CoV-2, all the respiratory infections, whenever you talk or sneeze or cough, the droplets you are expelling contain viruses in them. Same thing with feces. Feces, if you're infected with poliovirus, your feces has tons of poliovirus in it. And you may wonder, well, how does someone else get that from me? People don't wash their hands, okay? They go to the bathroom and they walk right out. And they have some fecal contamination on their hands. You can't see it. And then they shake someone else's hand, or they touch something and someone else touches. That's how poliovirus is transmitted. Right? And same thing with norovirus, fecal contamination of hands. You know, those mints in the restaurants by the cash register, don't touch them. They are contaminated with E. coli, yes.
Can a virus be both, two things? Would HIV be like germline and vertical?
So HIV does not infect the germline. Fortunately for us, right? It only affects T cells and some other cells, but no germline oh boy, that would be a killer, right? Because we can't get rid of it as it is from latent memory T cells. And if it were in the germline, good thing it can't infect germ cells, yeah. But you don't always need shedding to transmit, okay? So these are some examples of ways to transmit. So the blood supply, right? If you have a contaminated blood supply, you give people viruses in the blood, you're transmitting virus. Intravenous drug use can transmit infection without shedding. Insect vectors, germline transmission, and vertical from mother to baby via the placenta. No shedding involved. So respiratory shedding involves droplets that you produce when you sneeze, when you cough, even when you talk. We're making droplets of different sizes. There are big droplets and intermediate size and small ones. And the big ones drop right to the floor in front of you. And so they're not likely to infect anyone. But some of them travel longer distances. You can see they're small droplets, 3 to 5 feet. So this is where the 6-foot distancing came into play. But it turned out that some SARS-CoV-2, 6-foot distancings for SARS-CoV-2, some SARS-CoV-2 particles are also in these very small droplets that can go long distances. And one of the reasons is that when you first expel a droplet, it quickly evaporates and gets smaller, so it can travel longer distances. So these droplets are a major way of spreading many viruses, but other viruses are also spread by what we call fomites, contaminated surfaces or objects. You have nasal secretion, you touch your nose, you touch the subway pole, you shake someone's hand. Ian Lipkin, my colleague, said he stopped shaking hands. He just does a hand bump and he eliminated all his colds. So I try to hand bump, but sometimes I forget. And a hand bump's not going to help me here, right? If you guys are shedding virus, it's still going to infect me. The subway pole is a disaster. Do not touch it. If you do, don't touch your face until you wash your hands, okay? You have to. I wear gloves until I can't take it anymore. It's too hot, so that I'm not touching my face with the gloves. But these are not all viruses. So SARS-CoV-2 turned out to not be transmitted by these touch thingies. It's mostly the respiratory droplets. Okay. Now, an important consideration is, which I mentioned earlier, is whether you are shedding during the incubation period. And this differs for different viruses. So we have 3 viruses here to illustrate this. And the incubation period is in blue. So there it is for SARS-CoV-2. And here it is for SARS, the original SARS, and influenza. So let's take SARS-CoV-2. This is an early in infection. It's a longer incubation period than we see now. But here it is schematically. And then the symptom onset, right? The end of the incubation period is symptom onset, symptomatic period. This is shedding of SARS-CoV-2, that blue graph. You are shedding during the incubation period. You have no symptoms. So you're walking around. This is why it transmitted so well. So many people, first of all, many people were asymptomatically infected, like 20 to 40% had no symptoms, but also you shed during the incubation period, and that's the killer. Compared to SARS 2003, there is no shedding during the symptomatic period, the pre-incubation period, you can see here. And the peak of shedding occurred at the peak of symptoms, and most people were really sick, they were in the hospital, and it was easy to contain infection spread. So that virus only infected 8,000 people worldwide. There was no community spread like SARS-CoV-2. That's the killer with this virus. And finally, seasonal influenza, there is a little bit of shedding during the incubation period. It's a very short incubation period, but still people can walk around without symptoms and spread viruses. So this is a really important consideration. Ebola virus, by the way, is not transmitted during the incubation period. So you could fly to the US, get infected in some other place, fly to the US, land somewhere, and then you get sick, then you're shedding, but no one has any clue what you got, right? Until they get a history. Oh yeah, I was in West Africa where there's an Ebola outbreak. This is 2015. So a guy flew from actually Nigeria to Dallas. He ends up dying. He infects a couple of nurses because they don't know for a while what he has. So it's a real problem. You don't know on the plane that they're infected. Sorry, which statement about transmission is not correct? All infections are transmitted by shedding. The route is determined by the site of virus shedding. Transmission is required to maintain a chain of infection. Speaking can produce an aerosol that can transmit, and horizontal transmission is among members of one species, which is wrong. Remember that? I don't know if you remember, but there was a doctor who was working from the U.S. He was working in Africa during the 2015 Ebola outbreak. He comes to the U.S., he goes to Brooklyn, he goes bowling with his friends. The next day, he runs a fever. They take him to NYU, and he's got Ebola. And so they wanted to sue him for endangering people. And I said to the lawyers, she's not shedding during the incubation period. Until he got that fever, he's harmless. So it never went to trial because those are the facts. The lawyers are really just, sorry if anyone wants to be a lawyer, but that's what RFK Jr. is. He's a litigation lawyer. He's an ambulance chaser. He goes and he consults for companies who want to sue vaccine manufacturers. So he's currently to sue over HPV vaccine. Because it says it isn't safe, it hasn't been tested properly, which is all nonsense. But he makes millions of dollars a year doing this. I don't know if you remember during his confirmation hearings, Elizabeth Warren said, "Are you willing to give up that income?" He said, "No." When I was young, if you did that, that would be the end of your candidacy. Not anymore. All right, most of them are transmitted by shedding is not wrong. It's not right. The others, the route is determined by the site. Transmission is required to maintain a chain. Speaking can produce an aerosol. Horizontal is members of one species. That's all correct. All right, to end up here, some other things that influence transmission. Geography, where you are, because there may be a specific vector or animal that is needed. And so we're going to look at an example, chikungunya virus. Chikungunya virus, an enveloped alpha virus transmitted by Aedes aegypti mosquitoes. You get infected. Anybody ever have never mind, don't answer. You know it because it's like dengue. You get joint pains, you get a rash, you get fever. It's just nasty. So originally, it was in Asia and Africa, right? The blue. Never anywhere else. 2004, it spreads to India. And then in 2007, it goes to Europe, Scandinavia, and Australia. What's going on here? These recent outbreaks are spread by a different mosquito, Aedes albopictus. It's shown there. And the virus didn't really grow very well in this mosquito before, but it sustained a 1 amino acid change in the viral glycoprotein, and now Aedes albopictus is a great host. And that explains the expanded range, because A. albopictus has a greater range than Aedes aegypti. So here's the situation in the US. This is the range of Aedes albopictus, all right? And it's expanding with climate change. Eventually, it's going to be the entire country. Before 2006, they came in with travelers, very rare. And then until 2013, we'd have so many cases a year from travelers. And then in 2013, we had local transmission in Caribbean countries and territories, which means that mosquitoes are spreading it to people locally. And then 2014, we had transmission in Florida, Texas, Puerto Rico, and the Virgin Islands. So we're starting to get local transmission from person to person by Aedes albopictus, and this is going to increase. And that's why the FDA just approved a chick vaccine. So we'll see how that works. And finally, the season matters for a virus infection. We're in the respiratory virus season. It's winter. You usually don't get colds in the summer, although you can, but it's much lower than at any time. So if you go in our summer to the southern hemisphere where it's winter, you will find colds down there. So here are some seasonalities for different viruses. Here's rubella. Number of cases with the time of year. You can see a very distinct seasonality. Influenza is a winter disease in temperate climates. And poliomyelitis also, in fact, varies by latitude. You can see different latitudes have different peaks of poliomyelitis. Why is this? It's not clear. For influenza, at least, humidity and temperature seems to make a difference. So in the winter, we have pretty low temperatures, we have low humidity, and that seems to favor the viruses being in very small droplets that can go long distances. But, there are also flu outbreaks in the tropics, and they're seasonal, so there's certainly no low humidity there. So we really don't understand most of this seasonality, but it certainly does exist, as you can see in this chart. All right, so now we have entered the host, and next time we're going to start considering the immune responses to infection, and we'll start with the earliest, the intrinsic, and the innate defenses.